I'm excited to keep the fungal theme going and to tell you about the work that we've done over the past, really, I think 12 to 13 years, actually, trying to really develop this system to study microbial interactions. And over the past year, I've actually been wrapping up my lab at UC San Diego and transitioning full-time to a position here at Arcadia Science. But what I'm going to tell you about today is some of the exciting results that and kind of themes that would have come out of the research from my lab over the past several years. Okay, so why did I get started studying cheese? Well, I entered the field at a time when it seemed clear to me that we really needed ways to be able to connect microbial ecology and the underlying mechanisms that are happening within communities. So as you know, we have very powerful ways to study microbial communities in situ, but it can often be challenging to then bring those communities in the lab, let's say isolated as pure cultures, and then study them in vitro in ways that recapitulate what's happening in the environment and really get down to understanding mechanistic underpinnings. And so I was drawn to cheese, not because I was a food scientist by training. I'm actually a E. coli geneticist by training, but it seemed to me like a really interesting level of complexity for a microbial ecosystem. The communities of cheese that we study are the communities that form on the surface of the cheese during aging. So these are called the rind communities. These are biofilms that grow on the wheel of cheese in a cave or cave-like environment during the aging process. And depending on the way the cheesemaker makes and ages the cheese, you'll get different types of microbial communities. You can just see by eye how different they are assembling. These communities are relatively simple. They form in a highly reproducible manner. They develop over the course of weeks to months, so on a reasonable timescale. And every time a cheesemaker makes a cheese, they make hundreds of wheels. And so we have lots of replicates. So it seemed to me like a compelling option for a system that we might be able to get some experimental tractability out of. So the question that we had when I started the lab back in 2010 was, can we actually reconstruct and deconstruct cheese? So can we bring the system into the lab? And we did this over the course of basically the first 5 years of my lab, where we went out and mapped the diversity of microbial community, successional patterns, geographic patterns across hundreds of different types of cheese around the world. We then took those, subsetted those cheese samples and showed that we could actually deconstruct these communities into their individual members. So we could create culture collections that were representative of all of the major genera that we found based on sequencing. And then we set about reconstructing communities in the lab. And so we developed an in vitro cheese-based system. So we take freeze-dried cheese, we autoclave it with water and agar and salt, pour it into petri dishes, and then we pull from our culture collection and mix together whatever combination of organisms and inoculate them onto the surface and allow them to form biofilms on the surface of these cheese-like plates. And so we were, I would say, surprisingly successful at doing this, given how challenging this sort of thing can be in most more natural microbial ecologies. So when we got to this point, we were then kind of, I think, well set up to start trying to tease apart what's actually happening, what's driving the formation of these communities. And I think one of the nice things about a system like this, where you can have access to both the in situ system and an in vitro system, is you can really throw a lot of approaches at the system at once and use them to leverage different types of the different approaches and glean different insights. And so a lot of what we've been doing in my lab over the years has moving, in top-down ways from the in situ microbial ecology, using things like metagenomics to make hypotheses that we can then potentially test in vitro. And then using the in vitro system, make observations, measurements, understand mechanisms, and then use that to give us an understanding of what's actually happening in the real world. For today, I'm going to focus on the bottom-up approaches. And I'm going to focus on a series of studies that we've done in the lab where we're really trying to get at the genes that are involved in species interactions. So, like I said, at heart, I'm a geneticist. I really want to understand how things work at the genetic level. And studying interactions and the genes involved in interactions is incredibly challenging. So we wanted to see if we could take some creative approaches using this in vitro system that we had developed to get at what are some of the genes that are involved in species interactions. And the approach that we ended up using was borrowed heavily from Adam Arcadia and Adam Deutschbauer labs based on their RBT-and-seq technology. So RBT-and-seq, as probably many of you know, generates libraries of randomly barcoded transposon insertions within a given strain. And what we ended up doing is we actually just used their E. coli library as a type of sensor. And I'll describe a little bit more about how we do this in a second. But the basic idea is that we thought we could take our in vitro communities based on, let's say, a camembert, and introduce this E. coli library. And the readouts of the gene essentiality from the RBT-and-seq experiments might tell us about the conditions that are present within the microbial community. Okay, so how does this actually work? We take, let's say, our starting E. coli RBT-and-seq library. So again, this is a collection of strains that have mutations at random places throughout the genome, and each mutant is tagged with a unique barcode so that we can track its frequency in the population. Okay, so we take our initial population, we measure the distribution of mutants within that population. Then we can take that E. coli library, just grow it on our cheese alone condition, so just sterile cheese-based media. And you can imagine that mutations in certain genes would make it impossible for E. coli to grow in this environment, and those cells would die and then drop out from the population. So you would see a loss of those barcodes in growth on cheese alone. So that would tell us something about what are the pathways perhaps that E. coli needs to grow on cheese. But then what we did was we now take that library and grow it in the presence of other species, either individual cheese isolates or combinations of species. And then we measure the barcodes again and look at the differences. And what we can do is compare across all these conditions to infer something about the requirements. So for example, this particular insertion associated with the barcode, this orange barcode, is lost in both of these alone and with partner condition. This means this gene is absolutely required to grow on cheese no matter what. We have cases where the barcode is present in the alone condition. So if you knock out the gene in E. coli, it doesn't seem to matter when it's growing by itself. But in the presence of the community, now that gene is required. Okay, so that's the interaction-induced requirement. And then we have this other category of genes where the mutant doesn't grow well on its own. However, in the presence of the community, now it's rescued. So it's this something about the interaction has alleviated the fitness effect of this mutation. Okay, so that's the general concept. Now, the first place we applied this was with in vitro camembert. So camembert is one of the simplest types of cheeses, microbiologically speaking. It has one main bacterial species present in the rind, Hafnia alveae, which is a gamma proteobacterium, and two fungi, Geotricum candidum, which is a yeast species that grows in a filamentous manner, and Penicillium camemberti, which is the main fuzzy white mold that you see growing on brie and camembert cheeses. So we reassembled our cheeses in vitro and then did these experiments. Okay, and so what we can see is when we just measure the E. coli sensor library alone, we can get the fitness for every single mutation in the E. coli genome that's represented in this population. So what you're looking at here on the x-axis is the fitness effect. So increasing negative numbers to the left means it has a stronger growth defect. then we can look at what the difference is when the sensor library is grown in the presence of the Camembert community. Okay. So in the presence of the community, there are still a large number of genes that have a fitness effect, but if you actually do the Venn diagram of these genes, there's actually only 8 that have core requirements. So they have a fitness effect in both conditions. There are 37 genes that only become important when the community is present. So these are these interaction-induced effects. So something about the presence of the community now makes these genes, when you're missing them, have an effect on growth. And then the largest set of genes are actually these community-alleviated genes. So cases where when E. coli is growing by itself, if it has a mutation in any one of these genes, it is not happy. However, in the presence of the community, now it's growing just fine in the presence of this mutation. So what's going on here? So we took a look at these gene sets, did mapping back to KEGG pathways and functional adaptation to try and figure out like what's actually happening in the presence of the community. And it turns out that the biggest signal that we got was for amino acid biosynthesis. And so what I'm highlighting here in green are all the genes where we had mutations that had a significant fitness effect when E. coli is growing by itself, but then were alleviated in the presence of the community. So what that tells us is that if you knock out the biosynthesis of any one of these amino acids, E. coli does not grow well on cheese, which is interesting because cheese is largely protein. But what that suggests to us is that E. coli can't actually use the protein around it. Maybe it doesn't have the proteases to break it down into usable chunks. And so when E. coli is growing by itself on cheese, it actually needs to make its own amino acids, because if you knock any of these out, it doesn't grow. However, in the presence of the community, now these genes are no longer required. So you can mutate them and E. coli will grow just fine. So what does that tell us about what's going on? But what that suggests to us is that there's actually fungal bacterial cross-feeding happening. So we know that fungi are really good at secreting proteases. This is true in cheese as well as many other environments. They're great at breaking down large macromolecular substances like proteins and carbohydrates. And so what we think is happening is that and we did this by further going down and like doing pair by pair assays. What we see is the fungi are secreting proteases that break down casein protein into usable bits so that E. coli can actually grow. And so by doing these genetic screens in different combinations, it actually revealed to us that there are lots of opportunities for cross-feeding apparent in this environment. Now, the other thing that we found when we started doing these different combinations of organisms is that these interactions are not simple and pairwise. In fact, about half of the genes appear to be subject to higher order effects. And so let me just walk you through one example here to illustrate how this works. So again, this is a map of the E. coli fitness distribution of these mutants. And there are these 2 genes here, ACRA and B, which in E. coli form this large molecular complex of an efflux pump for antimicrobials. When E. coli is growing alone on cheese, if you knock these out, it's fine. It doesn't care. E. coli will grow just fine. However, in the presence of Geotrichum, now these genes become important. So that suggests to us that Geotrichum is producing an antimicrobial in addition to producing proteases that make it so that E. coli needs to be able to efflux antimicrobials or else it has a growth defect. However, when you now grow the whole community together, these genes now become non-essential again. So what's going on here? This suggests to us that there is higher order interactions happening, that the combination of multiple members of the community is changing the effects on the fitness. Okay, so what I've told you is that Geotrichum is secreting proteases, which is allowing E. coli to cross-feed on proteins and amino acids. However, it's also secreting antimicrobials, which requires E. coli to produce efflux pumps. However, what we found again through going back one by one is that Hafnia, the other bacteria in the community, is actually able to cross-protect E. coli. So Hafnia, we think, is producing some sort of antimicrobial resistance factor that it secretes into the community. And the presence of Hafnia actually makes it so that E. coli no longer needs its efflux pumps. Okay. So multi-layered interactions, even in this very simple community. Okay, so where do we go next? So this started to give us hints that fungi are doing really interesting things in these communities. They are producing things that are stimulatory and inhibitory at the same time. And so we were really motivated by this, and also studies that had come out of Ben Wolf's lab. So one of my former postdocs, who's now at Tufts, who's been expanding a lot of the work into fungal interactions in cheese. And it just seemed like fungi were really the key players in this ecosystem. And so one of my first grad students, Emily Pierce, decided to take on as her thesis project the challenge of trying to map out the diversity of effects that fungi might have on bacteria in the system. So what she did was she assembled a panel of different fungi, yeast and filamentous fungi, and she combined them with our interaction sensor library again. And by doing this, she could then look across all of these different fungi and categorize the effects on different pathways. And so again, you're looking at these fitness diagrams where we have the fitness of the E. coli gene alone on the x-axis now, and the average fitness with a fungal partner on the y-axis. And so the thing that stands out, hopefully here, is that there's this cluster of pink mutants, largely FEP genes. And these FEP genes are important for iron uptake, and I'll describe more about that in a second. But it seemed like an effect that was present across all of the fungal species we tested. So the fact that these circles are large meant all of the fungal species were having the same effect of alleviating a fitness effect of iron uptake mutants. There's also many other things going on here, which I'm not going to talk about today. And just to show you maybe a little more clearly, if this is sort of the wild type growth level at the top, and in the open circles down here, this is the fitness effect of the E. coli mutant growing by itself on cheese. Now you can see the improvement in the presence of any of these other fungi. So all of the fungi seem to be improving the growth of these particular mutants. So what's going on here? So again, you probably all know about siderophores, and we often think about siderophores in the context of bacteria. And bacteria, we know, use siderophores to get iron from their environment. This is true in cheese as well as in soils and many other environments. Now, the process of getting siderophores is multifaceted. You have to produce the iron chelator molecules through often complex biosynthetic pathways inside the cell. You have to produce exporters to pump them out where they chelate the iron. And then you have to produce uptake systems to pump back in the chelated iron. Now, what we were seeing were defects in the uptake systems being really detrimental and then being recovered in the presence of the fungi. So this made us think that maybe there was something going on where maybe the fungi were actually supplementing or somehow allowing uptake of siderophores when E. coli was missing the genes to do this. And like I said, usually we think about siderophores in the context of bacteria, but it turns out if you do some very cursory searches, people have known about fungal siderophore production for a very long time. They've known about siderophore production in fungi from cheese, actually, for a long time. And interestingly, it's been known for almost an equally long time that E. coli produces a fungal siderophore uptake pathway. In fact, it produces a couple different ones. Why it does that, we don't know. There hasn't been a lot of work on the ecology of this system, but these systems have actually been very well characterized. So what we think is actually happening here is that the bacteria are stealing fungal siderophores from the cheese fungi. So what our genetic analyses have told us is that if you get rid of the FEP system genes, E. coli doesn't grow well on cheese. We know cheese is iron limited, so that means they're not able to take up their own siderophores anymore, so they can't grow. However, in the presence of cheese fungi in the community, the fungi are also producing siderophores because they also need to take up iron. And these get chelated, and actually E. coli produces the FEW system is one of the fungal uptake systems that it produces. We've showed genetically that this is the one that is being recognized in this case. This system is now required to allow the E. coli to take up the fungal siderophores. Okay, and this was true across all of the fungi we tested. And we also tested some fungi from skin and soil to see if there was a similar effect, and they could also rescue these FET mutants. So this seems to be a widespread phenomenon where bacteria will actually cross-feed siderophores from neighboring fungi. Okay, so the last little bit, I wanted to give you a quick preview of a story we've been working on for the past couple of years about phage and how this actually brings us back to fungi as well. So for a long time, we sort of prided ourselves on the fact that, okay, we weren't just studying bacteria, we were studying the bacteria and the fungi, and fungi are so understudied in microbial ecosystems. So, you know, good job us. But then we sort of realized, well, actually, we're ignoring another major component, which are the viruses in the system. So we started to get really interested in characterizing the phage and understanding what they were doing. So one of my grad students, Tara Spencer, decided to take this on as her project and see, can we actually isolate phage from cheese? And indeed we can. And in this in vitro camembert model, using Hafnia alveae as a host, we can isolate at least 2 major types of phage from the system in quite high titers often. And so she set out to use this genetic approach, but in one of our native cheese bacteria. So we generated a RB10-seq library in the cheese Hafnia. And then we did the same types of experiments that we've been doing, but with the in vitro camembert community with the Hafnia library instead of E. coli library. And so basically we did all these different combinations of in vitro camembert plus and minus phage to see what the fitness effects were and what the general effects were on the community. And we found something, I think, which is really interesting. What we found is that you can see in all the solid lines here where we don't have phage, you can grow the Hafnia library alone, grows just fine. You can grow it with the different fungi. They all grow together just fine. We knew that from, you know, the wild type studies. However, when you start introducing phage into different community combinations, you see very different effects depending on who's there. So if you just grow the phage with the Hafnia, which is TS33 is the Hafnia alveae strain, you can see an immediate drop in the Hafnia culture, but then you get regain and then some drop again. So there's some sort of resistance to the phage happening here. However, in the presence of any of the fungi, we don't get this phage resistance spike happening. In fact, what happens is the Hafnia dies out after a couple of days. So what's going on here? Well, what we think is happening is that there's a trade-off going on. And what we found by looking at the mutations that are present in the RBTN-seq library of these Hafnia populations is that you end up getting phage-resistant mutations in the cell envelope, which is, in this particular case, we see a lot of LPS mutations. We think this is a phage that binds the LPS of the cells. And basically, if you knock out these genes, you get resistance to the phage. However, if you knock out these genes, you also get increased susceptibility to fungi. So if you remember, these fungi we know are producing antimicrobials to some extent, and we know that cell envelopes like LPS are important for cellular stability, integrity in the presence of antimicrobials. So we think what's happening is that the bacteria, if they are forced to become resistant to phage, that makes them it makes it really hard for them to grow in the community because they can no longer resist the fungal antimicrobials as well. So what we think this is telling us is, again, multi-layered interactions happening even in this simple community. So if you have a population of Hafnia where most of them have their intact LPS, but maybe you have one that has a mutation in the LPS, that one that has a mutation is resistant to phage, so you will get resistance arising from now outgrowth of that resistant mutant and death of all of the otherwise wild type cell envelope cells. However, once that population grows up, it now is actually more susceptible to fungi and to potentially to antimicrobials they're producing. And so I think this is just a really fascinating example of an ecological trade-off that we can show in a very simple community that is being played out by interactions mediated through phage and fungal species with bacteria at the center of it. Okay, so hopefully this will drive home the message that we think fungi are really key players in this particular microbiome. And I don't think that any of these lessons are specific to cheese. I think these same sorts of things, cross-feeding, competition, antimicrobials, are going to be happening in many other microbial ecosystems. So I think we always need to consider fungal or microbial eukaryote members when we're thinking about communities. I think one of the fascinating things is that because the fungi have large genomes and they have immense biosynthetic capabilities, they have the ability to secrete this array of potentially stimulatory and inhibitory molecules at the same time. And by doing that, they can really shape the types of membership of the surrounding community. So to summarize, we've taken this approach of adapting genetic screens for studying interactions in these in vitro communities. We've done this in a way that I think has highlighted a lot of interesting biology that can be translated to other areas. And then I think, one take-home message that I didn't really talk about is, a lot of what we can infer here based on what's going on is based on what we already know. We know a lot about siderophores, antimicrobials, but over 50% of the genes we identify as important in interactions have completely unknown functions. And so there is still a huge amount of biology underlying interactions in communities that we just, even with the genetics, have a hard time accessing. So lots of room for future work here. And with that, I will just thank, this is my former lab at UCSD, who are all off doing cool, exciting other things now. And with that, I will take questions, and thank you for your time.
Great. Before we proceed with questions, I encourage everybody to stay for a couple more minutes. I have two important announcements. With that, any questions for Rachel?
Great talk, Rachel. I really enjoyed it. It's going to be a loss to the community for you to no longer have an academic lab, but I'm sure Arcadia will benefit mightily. I'm curious about the fungal siderophores that E. coli is taking up with the foo system. Are all fungi making the same siderophore, or are different fungi making different siderophores in this system? And is it known what those siderophores are?
Yeah, we've collaborated for a long time with Laura Sanchez's lab, who's at UC Santa Cruz now, on trying to characterize the siderophores. In some cases, they're making the same siderophore, so there's like coprigin type compounds, but other cases they're making siderophores that we haven't been able to identify yet what they are. So yeah, still I think a lot of diversity there of fungal siderophores to be explored.
Any other questions?
Hey, Rachel, thanks. That was a great talk. I was curious about the antimicrobial part, which you brought up twice with the Hafnia. And I was interested because the first time it seemed, I was envisioning a secreted antimicrobial, antimicrobial that the Hafnia was secreting something to degrade it because it
Yes.
helped out the E. coli. But then in the second part, you were talking about a cell envelope mutation. And I was wondering if you thought about how to reconcile that, or is Hafnia doing two things? Is it also degrading the antimicrobial?
Yeah, a great point. We have not been able to track down what the exact antimicrobial is. Again, like collaborating with chemists, and we also collaborate with Nancy Keller's lab to look at the LA mutations in this fungal background to narrow down, like, different types of biosynthetic pathways. We don't know what the antimicrobials are that the fungi are producing yet. And in terms of Hafnia, I think it's very possible that they're producing antimicrobial resistance factors. So, it's not a beta lactam, but let's just say it was a beta lactam, maybe secreting a beta lactamase or something like that. But then in addition to that, they do still need an intact cell envelope to, protect them. So I think there's, they could be overlapping or complementary effects, or they could be distinct pathways. Maybe they're even operating through distinct antimicrobial type processes. So